Hello, my dear O level students. In our last videos of microorganisms and biotechnology, we were talking about viruses, we were talking about bacteria, and here in this video, we are going to talk about fermentation. We need to talk about the design of fermenter, and we will also talk about the ethical concerns of bacteria and why bacteria is preferred to be used in genetically modified products. So, here on your screen, the objectives ask us to discuss why bacteria are useful in biotechnology and genetic modification. For this, let me give you a little example. Consider we need to make insulin. For example, there is an inquiry in which you may need to make insulin. So, what you do is you extract DNA from from the pancreas. This is your pancreas, and this is a gene for insulin production. Now, cutting a long story short, what we do is we take a bacteria and we know there are plasmids inside the bacteria. Apart from the large circular DNA that is present inside bacteria, we know that we have plasmids. So this one is a plasmid. Now what we do is we extract plasmid from here. So let me make a larger plasmid here. We cut it from this side, and then what we do is we paste this insulin gene inside the plasmid. So now this plasmid contains insulin gene. Now when this plasmid is containing an insulin gene, so this plasmid is said to be a transformed one. It's a transformed plasmid. So when we introduce this plasmid back into this bacteria is a transformed bacteria. It is said to be genetically modified bacteria. So first this plasmid is going to increase its copy number inside this bacteria. Copy number means number of plasmids. First it is going to increase its copy number, and then when this bacteria will grow, bacteria, if you provide them with all the suitable condition, it takes 20 minutes. It takes 20 minutes uh, to replicate a bacteria, to replicate one cell into two. So they have a very short generation time. Their generation time is very short. And since in nature bacteria love to share plasmids with each other, it's very easy to introduce a transformed plasmid back into bacteria. So it is very easy to be manipulated. That's why scientists, they mostly use bacteria for genetic manipulations and for genetic engineering experiments. Because here this plasmid acts like a vector. So once we have transformed bacteria with us, what we need to do is we need to rear these bacteria. We need to give them nutrients and all the good conditions that they deserve, that they need, and in return, they are going to produce insulin for us. Now let's talk about the structure of a fermenter. Fermenter, also called bioreactor, bioreactor or fermenter. It is such a vessel, it is such a machine in which microorganisms are cultured, in which microorganisms are reared. So let's see how these microorganisms are cultured inside this bioreactor or a fermenter. So first, I want you guys to tell me what are the requirements for rearing or for having bacteria. अगर आपने bacteria पालने हो, what do you need to maintain? Number one, you need to maintain suitable temperature. You need to maintain suitable pH. You need to give them nutrients. You need to give them water. You need to give them security. And you need to give them oxygen. So if you're providing all of these things to a bacteria, they will grow and they will start producing your product of interest. So let's see how these things are managed inside a fermenter. So for this fermenter, we have a large vessel that is enclosed. But definitely it has some openings. All right. Here we have a sparger, which is connected to a filter, and then it is connected to a pump. This is a pump. This is filter, and this is स्पार्जर. इसमें से हवा आती है. क्या आती है? हवा आती है. और छोटे छोटे बबल्स की फॉर्म में ये हवा आपके फर्मेंटर के अंदर आती है. सो पहली रिक्वायरमेंट जो हमारे पास ऑक्सीजन की थी प्यारे बच्चों, वो हमने कंप्लीट कर ली. सो हम इसको हाईलाइट कर लेते हैं. नाउ वी हैव सक्सेसफुली प्रोवाइडेड देम विद ऑक्सीजन. सेकंड थिंग, सिक्योरिटी. We want to maintain aseptic conditions. Having an enclosed vessel makes sure that no other bacteria is there along with our genetically modified bacteria. So, having this large vessel ensures this. Then, we need water and nutrients. Now, what we do is we fill up this fermenter with these bacteria and nutrients and water. Definitely, everything is dissolved in water here. So, it's quite easy to understand. So, water and nutrients. Nutrients are dissolved inside. Now, we need to maintain temperature. In order to maintain the temperature, we have a temperature maintenance jacket around it. From one part, there is an inlet of water, and the other part, there is an outlet of water. Now, we have temperature probes which are attached to automatic computer, and you can watch on the screen whatever is going on. If the temperature is low, so this is water inlet, and this is water outlet. This is water inlet and this is water outlet. अगर इसका overall temperature low जा रहा हो, it starts adding hot water, warm water around the fermenter, so that constantly एक तरह से heat इनको provide हो जाए. और अगर इसका temperature up जा रहा हो, तो it starts putting in cold water inside. Then they also have pH probes and suckers and buffers. So we have alkaline buffers and we have acidic buffers. If the pH of system is going beyond the set range, then suckers automatically suck uh, acidic or alkaline buffer to maintain the pH of the whole system. Then we have a shaft, a motor, and a rotor to which small impellers are attached. So these are called impellers. This is a motor. This is a shaft. And these are impellers. What these impellers do is they keep on moving, moving, moving so that all the contents they are thoroughly mixed inside, right? Acha. Then here we have an inoculation chamber. So here we have inoculation chamber. पढ़ाते पढ़ाते नींद आने लगी है इसलिए मैं पढ़ा पढ़ा के थका हुआ हूँ. अच्छा, अब हम करते क्या है? व्हाट वी डू इज, ठीक है, अब हम करते क्या है कि हम इसके अंदर अपने न्यूट्रिएंट्स डाल लेते हैं और इसके अंदर अपना जेनेटिकली मॉडिफाइड बैक्टीरिया, जीएमओ बैक्टीरिया हमने इसके अंदर इंट्रोड्यूस करवा दिया और यहां से इसका आउटलेट है. We wait for a good period of time and providing all the good conditions to bacteria. Now we are expecting that this transformed bacteria that was carrying an insulin gene will start producing insulin. And once this batch is complete, once we are done with this batch, then what we can do is we can extract all the contents which were here and then we can extract insulin from that material. Now let me show you picture of an industrial fermenter. Look at it. These are pilot scaled but industrial fermenters. So they are quite complex. They have involved a lot of research and development. But still, आपके लिए ये वाला fermenter अच्छा है, प्यारा सा, छोटा सा, नन्ना मुन्ना सा, because at least you understand whatever is there inside. So स्पार्जर का काम है ऑक्सीजन देना, टेम्परेचर जैकेट का काम है टेम्परेचर कंट्रोल करना. उसको एयर टाइट इसलिए बनाया जाता है ताकि कंपटीशन फ्री हो जाए. पानी में न्यूट्रिएंट्स ऐड होते हैं तो वो उसको मीडियम के तौर पर मिल जाता है और बैफल्स घूमते रहते हैं ताकि उसके अंदर किसी चीज की सेटलमेंट ना हो जाए. सो दिस वाज अबाउट यूज ऑफ बैक्टीरिया इन बायोटेक्नोलॉजी एंड फर्मेंटेशन. So in my next video, inshallah, we are going to talk about food biotechnology. So stay with us. Take care of yourself. फिर मिलेंगे.